So that's it for my practical announcements, and that means it's time for our first speaker, Sofie Derycke from ILVO, who will present the main findings of the pilot studies and the recommendations for using DNA-based methods in marine environmental monitoring. So Sofie, the screen is yours.
Okay, thank you, Louise. And also from my side, welcome to everyone. It is with great pleasure that we present today the main results of our pilots in this first presentation. And let me start with introducing why we started GEANS almost 5 years ago. It is clear that our seas and oceans are experiencing quite some pressures from human activities. For example, offshore wind farm construction, fisheries-related activities, sand extraction, but also indirect effects of human activities like climate change, and as a result of that, the increase of the surface water temperature of our seas and oceans are impacting the marine ecosystem. Of course, we want to keep on exploiting our system, but we don't want to end up with a system that is completely devoid of animal life, as seen here in this picture. We actually want to have a balance between the human activities and a lot of species being present in our system. A way to do that, to achieve this, we need fast and accurate monitoring techniques. And already before GEANS, several studies indicated that the use of DNA with metabarcoding or DNA can be a very effective means to do this. So the focus area of GEANS was the North Sea, of course, because it's an Interact North Sea region funded project. And as Louise already pointed out, all the countries surrounding the North Sea were included in the GEANS consortium. And our main focus was actually on macrobenthos, so the invertebrate taxa that are relatively big, so their body size is larger than 1 millimeter, and that live in or near the seafloor. Now, when we talk about DNA-based methods, it's important to make the distinction between what we call bulk DNA and environmental DNA. And to give you a very broad explanation, if we are using bulk DNA, we are still extracting the animals from our sediments, from our environment. We mix them into a homogeneous solution, and this is then used as a template to further assess diversity. In the case of environmental DNA, we look at DNA molecules, tissues, cells that are suspended in the water column or that are present in the seafloor, without extracting animals from the environment. So in this case, environmental DNA is a non-invasive method. Now, the heart of GEANS consists of stakeholder-driven pilots. So we chose these pilots in close collaboration with stakeholders. And the main aim of these pilots was actually to see whether these DNA-based monitoring approaches can be reliable, accurate, fast, and cheap for our monitoring objectives. In this respect, the pilots that were selected were already conducting monitoring with traditional morphological-based analysis for many years. We focused on soft sediment pilot studies, also on macrobenthos living on hard substrates. We also had a pilot on detecting non-indigenous species in harbors. And in an extension phase of the GEANS project, we also looked into the possibility of eDNA from the seawater for monitoring marine diversity. If I now zoom in a little bit into each of these different pilots, then the biggest pilot that we had was a soft sediment pilot. And this consisted of 4 subcases, 2 of them focusing on environmental impact assessment. So in Belgium, we analyzed samples, both morphologically and genetically, linked to sand extraction activity. In the Netherlands, the mechanical harvesting of lugworms was used as a test case for environmental impact assessment. In Germany, we focused on a long-term monitoring station for which over more than 45 years of monitoring data using morphological data is available. And in this case, subcase, 5 stations were sampled in 3 different seasons. Our last subcase within the soft sediment was conducted by our Danish colleagues in the framework of MSFD monitoring. And in a subset of the samples for this MSFD monitoring, our colleagues collected sediment to see whether eDNA from the sediment could actually represent the macrobenthos community in these samples. Important to note here is that a set of these samples were collected at a deeper zone of 44 meters, while the others were coastal locations. It became already very soon clear that DNA-based methods can not always provide what we hope that they are providing. Because, for example, in the case of the mechanical harvesting of the lugworms, we were not able to obtain any PCR products. We don't know why. There was a lot of testing in the lab, whether it was the fixative of the samples or the DNA extraction. At some point, something went wrong, which disabled us to get any results based on DNA for this subcase. Now, for the results of the 3 other subcases are shown very briefly on this slide. What we saw, for example, for the sand extraction impact assessment is that the number of species detected with our DNA-based method, which is indicated here with the striped bars, this DNA-based method detected more species consistently compared to the morphological analysis, which is indicated here in the colored bars. We also saw that these 2 genetic methods actually revealed the same ecological pattern, meaning that in the medium impacted location, the highest number of species was observed for both methods. However, if we look at the species that were detected, we see that approximately 1/3 of the species are shared between the 2 methods. And each of the methods was actually also picking up species that was not present by the other methods. We also saw that the main characteristic species explaining the differences between these impact zones were similar, whether we were looking at DNA-based analysis or morphology-based analysis. If we then look at a long-term monitoring station in Germany, we see that the number of species were very similar between the 2 methods, except for the summer period, where the DNA-based methods, here indicated in purple, detected more species than morphology-based methods. In agreement with the previous subcase, we see again that quite some species are shared between the 2 methods, but also uniquely detected species by either method are found. In this case, the species that were explaining the differences between the seasons were actually different when we looked at DNA or morphology. And then for the MSFD monitoring subcase, what we found is that the morphological identification, here indicated in blue for each station, generally picked up more species than the eDNA data. And we also see that the overlap between the eDNA from the sediments and the morphological identification was very, very low. As a result, the species characteristics for each depth zone were different between the both methods. Across all these 3 subcases, we also measured during every step the time and costs involved for the 2 methods. And what we see for each of the 3 methods is that DNA-based metabarcoding is actually faster and cheaper than morphology-based analysis. The second pilot that we conducted within the GEANS project was looking at hard substrate macrobenthos fauna. And for this, a well-established method was used, the autonomous reef monitoring structure, abbreviated as ARMS here, which is actually a 3-dimensional structure with different settlement plates on top of each other, which is deployed in the marine environments and for several months, so that the hardsup fauna can start to colonize these arms. These arms in the framework of GEANS were deployed in 5 different coastal locations by each of the partners. They were also collected by the different partners, but the analysis, the laboratory workflow, and the bioinformatic workflow of all these samples was actually done in a centralized way as part of a bigger project of MBOM. What these arms showed is actually that a lot of different taxa are colonizing the arms, and especially multicellular animals can easily be picked up by this method. Also, within these metazoa, there are a lot of different taxonomic groups. The main marine phyla are present there. And what we saw is that if you look at the species list, we can actually investigate whether non-indigenous species were present in these arms. So we see here, for example, in the different, in 5 different deployment sites, each of them contains NIS species. They also had sensitive species, meaning, for example, long-lived macrobenthos species that are very sensitive to changes and disturbances. And also a limited number of actually red-listed species was detected. So ARMS was actually a very standardized method allowing for large-scale comparisons of hard subfauna across different regions. Our third pilot was focusing on the detection of non-indigenous species in harbors. And depending on the country that conducted the case, the sub-case pilot, different ways of sampling this NIS was conducted. So mainly for morphological analysis, for example, grab samples from the sediment or zooplankton samples or settlement plates were used. If we then have a look at the different results from each of our partners, then we see, for example, in Belgium, where settlement plates and zooplankton samples were used, that actually the DNA analysis of zooplankton detected almost all NIS present. So in this case, bulk DNA of zooplankton seems to be a good choice to detect NIS. If we then go to our German colleagues, where they use settlement plates and grabs to sample this NIS, we see that bulk DNA morphology pick up a similar number of species, but also species that are uniquely for either method. So this means that if you want to detect all NIS species, that we would need to combine both the bulk DNA and morphology-based analysis. Our colleagues in Denmark conducted used settlement plates, but also looked at eDNA from the water column, and they compared metabarcoding with qPCR as a method to detect this NIS. Also here, the DNA-based methods detected more NIS compared to the morphology-based analysis. And in this case, also some species were uniquely detected by either of the methods. So again, here, if the main aim is to detect all species in your system, then a combination of both would be recommended. The bulk DNA method here was 20% faster, but it was more expensive than morphology-based analysis. This is in contrast with the numbers reported by our Belgian partner, where the DNA-based method was 93% faster than the morphology and 65% cheaper. And these differences in number reflects actually the expertise of the partners in hand, because of course, if you're not that well expert in morphological identification, it takes a long time to identify them morphologically, which biases these numbers here. I think these are the more realistic numbers in case you have an institute that is able to do both the morphological and the genetic analysis. The Sweden partner did look at eDNA from the water column and also bulk DNA from zooplankton samples. Again, here showing that the two methods are quite complementary in detecting the number of species here. And important to note here is that with the arms that were deployed, no NIS were detected with the morphological identification. Our last pilot was conducted in the extension phase of GEANS because more and more studies were showing the potential of eDNA for monitoring. So we decided to apply for an extension and see whether this eDNA-based monitoring could also be of use for the stakeholders that were already involved in the GEANS project. So we have 3 sub-cases within this eDNA pilot, where we focused on the impact of offshore wind farms in Belgium, where water was collected in concert with beam troll data. In the Netherlands, the focus was mainly on method optimization, where our colleagues designed a new primer set to amplify a much longer fragment, with the idea that the longer your fragment is, the higher the resolution becomes to distinguish different species from each other. This new primer set was used on a public aquarium because, of course, we know there which species are occurring. So it's a way of ground truthing our eDNA results. And our colleagues also applied this near shipwrecks and near offshore wind farms. And then the third monitoring subcase was again looking at NIS monitoring, using either settlement plates and seawater or zooplankton and seawater. So briefly reflecting on the results for the offshore wind farms, what we see is that for the fish species, and this was actually the only pilot where we were not focusing only on macrobenthos, but where we included both invertebrates from living on top of the seafloor, so the invertebrates that are caught with the beam trawl, but also the fish communities. And what we see is, as typically observed in eDNA studies, is that we pick up more species, indicated here in blue, compared to the morphological identification in different regions in the North Sea. Well, this was for the invertebrates less the case. So both eDNA and morphology were in many regions quite similar, although there were some exceptions also. Importantly, for the eDNA of the fish communities, we saw also nice congruence between the eDNA results and the morphology if you look at spatial distribution. So in this plot, the green samples are all samples collected in the coastal sites, and then the offshore wind farms are here indicated in purple and orange. And you see also in the beam trawl data that there is a nice difference between the coastal and the offshore communities. Also, if we zoom in within the offshore wind farms, we find differences with eDNA that are similar with morphology of the fish. Importantly, 83% of the fishes that were found in our beam trawls were picked up with eDNA. And this is in stark contrast with the only 27% of epibenthic invertebrates that were detected with eDNA. We also tracked time and cost for beam trawling and for eDNA, and beam trawl analysis is actually much faster and cheaper than eDNA metabarcoding. And this is, of course, mainly because the beam trawl analysis can be done immediately on board of the research vessel. For the optimization with a longer fragment, we noticed that this method was able to detect 58% of the fishes that were present in the aquarium. Also, if we apply this to the North Sea, a broad diversity of fishes were detected. But of course, this needs a little bit further ground truthing because a longer fragment also means that we need a reference database that is containing these longer fragments. So the 58% here is an artifact of the fact that many of the fishes don't have the long fragment already in the reference database available. So we need to keep investing in this reference database, and we need to compare these results with short read data to actually prove that this longer fragment would be able to detect more fish species. Then for the NIS monitoring pilot, we found that eDNA was able to detect twice as much species than the morphology-based DNA. And this is the same actually for the German pilot, where eDNA detected actually 4 times more species than the morphology-based analysis, but even using bulk DNA from the plankton net detected most of our species. So to conclude and to formulate briefly our recommendations, to answer the question, is DNA-based monitoring reliable, accurate, fast, and cheap? I think, as very often in science, the answer to this question is not that straightforward. And you see a lot of buts on this slide. There is definitely room to include this DNA-based monitoring, especially for the soft sediment monitoring case. So the pilot that we conducted there clearly showed that bulk DNA was much better than eDNA from the sediment for macrobenthos monitoring. We have validated this bulk DNA protocol. We have a curated reference database for the North Sea for macrobenthos, and we showed that it's faster and cheaper. And this was consistent on all the subcases that we conducted. So these are all very positive points of using DNA-based monitoring. Of course, sample preservation is key, as was demonstrated with a mechanical luckworm experiment, and which I did not have the time to include this in the presentation, but we also compared formalin fixative with ethanol fixative, because formalin can, of course, not be used for DNA-based analysis. We found very few differences between the two fixatives and would therefore recommend to switched to ethanol for preserving macrobenthal samples. Importantly, and this is true for all the case studies that we did, with DNA-based methods, we cannot provide information on numbers of specimens for each species. So abundance biomass information is not possible with DNA-based barcoding. Metabarcoding. We also don't have information on the life history, so we don't know whether there are juveniles, adults, eggs present in our sample. And of course, the species identity, as we showed, differs a little bit between the two methods. Also, at this point, there are no good indicators available to use DNA-based measurements to assess the good environmental status of the marine environment. So for the soft sediment monitoring, what we recommend at this point is to integrate bulk DNA and morphology. So not to just pool everything and do it twice, but integrate it to achieve the highest resolution possible to benefit from the positive points from either method without increasing the cost of this monitoring. For the NIST monitoring pilot, we saw that bulk DNA was actually, in all the cases that used bulk DNA from zooplankton, detecting more NIST species than eDNA from the water, and also with the morphological-based analysis. But each method was also able to detect unique NIST species. So if the aim is really to find all NIST present in your harbors or your environment, the best approach would be to combine everything. However, if your resources are limited, then a focus on bulk DNA from the plankton combined with eDNA, which is a limited extra cost here, this eDNA, would be the best choice. We want to highlight also that for NIST monitoring, the sampling design is very important. Because if you sample, of course, in a reproductive season, eDNA from the water may give you a very good result of detecting NIST. While if you would sample in a period where they are not reproducing, the eDNA may be less valuable. So taking care of this temporal and spatial replication is important. Our eDNA monitoring pilot showed that for monitoring fish with beam trawl data, that we could actually move on to eDNA from the water, just because it's non-invasive and non-destructive, and we get the general patterns of fish communities. However, for epibenthic invertebrates, it is not useful because we don't detect a lot of these epibenthic invertebrates, mostly because probably they are having these hard exoskeletons and just to release a very small amount of eDNA in the water. Keep in mind, of course, if we switch to eDNA for fish monitoring, that we actually don't know whether we are looking at eggs or adults, and that we also showed that it's not really cheaper or faster. The big benefit is that it allows us to increase the sample size to an extent that is just not possible with beam trawling, without a much increase in cost. But further method development for this eDNA is needed, is ongoing, and we need to harmonize this across countries. And then finally, for the hard sub monitoring, our ARMS studies demonstrated that this is a very easy and standardized method to inventory these hard subspecies, and that looking at DNA-based analysis for these ARMS allows to streamline and the sample processing, but also generating the species list. We need to take into account, of course, that these ARMS need to be deployed for a longer time because organisms have to have time to settle and grow there. And our time and cost calculation also showed that they were very similar compared to morphology-based. So here there's not really an advancement in time or costs when we move to DNA-based methods. So with this last slide, I hope I was able to give you a glimpse of all the different pilot studies that we conducted in close collaboration with our stakeholders, and that the DNA-based method definitely has a lot of advantages, but we need to be aware of the disadvantages of this method as well for adequate biodiversity monitoring in the North Sea.